So good morning, everyone. I'm just about awake. Are you? Yes. Some nods. Well, hopefully I can stimulate you to stay awake for a little bit longer until the coffee break. So you've actually got a slight double bill from me. So I'm going to first of all start off talking about the EBI's Metagenomics platform and some of the challenges and issues we have. So this is very much a generalist metagenomics analysis platform. And I'm going to talk about a lot of the aspects, not so much the website and interface, but some of the challenges we face and some of the things we found over the years of developing this resource. So hopefully everyone in this room is fairly familiar with metagenomics, and the sort of big three in terms of biomes that people have investigated are the human-associated microbiome, soil, and aquatic biomes. However, so I started at EBI in 2014, and even in that, since 2014 and running the resource, things have really changed in terms of the environments people are looking at. So it's no longer just those sort of big three that we see. We see all sorts of new things. So biofuel production and looking at fermentation and sort of decomposition, so that people go into compost heaps and try and find those cellulose degrading enzymes that, and then they try and recover those from metagenomics. So the oil field or oil industry, it's a massive industry, and who thinks that they're interested in metagenomics for sort of, you know, pollution point of view and sort of clean up? I've got a few nods. Who thinks they're interested in metagenomics for their own purposes? The people at the latter. So what they're really interested in are two things. So they're really interested in the biofilms that form on the inside of pipes at refineries, because what happens there is you get this film forming the inside and it causes corrosion, and if they get a break in that pipe in the refinery, stopping a refinery costs about $1 million a day. So they're really trying to work out how to stop these biofilms forming. And there's also things like oil field souring, where you actually get bacteria going into the oil fields, metabolizing the complex hydrocarbons and producing hydrogen sulfide, and to remove that from the oil is actually very, very expensive. And so they're trying to work out how to eliminate that using bio processes. To be honest, things like Deep Horizon, they're not so worried about. The other big area that's getting a lot of interest is sort of things like green chemistry. So green chemistry, this is a sort of buzzword in the pharma field, in the fact that to produce most drugs, they use very complex chemical synthesis with a lot of byproducts. And a lot of drugs are very unstable. But if you can get bacteria to produce that drug or other microorganisms, then you can deliver that to the point where that drug is needed, say Africa, and the cleanup is literally you boil the fermentation. So there's a lot of interest in all of these different areas. So we've got huge diversity in biomes that are studied. That's number one. And now I'm going to oversimplify the sample collection, because I'm a sort of dry lab person, so that's just a black box to me. So, you know, mid-2000s, really everyone was using size fractionation to pull out that sort of mainly bacterial component, and then they'd probably just do amplification. And I believe that you heard a lot about amplification yesterday or earlier in the week. However, now this sort of size fractionation is no longer the in thing. People tend to just take all of the microorganisms, because it's actually between the single cellular eukaryotes and the viruses, there's overlap in the size. You never get perfect separation anyway, and people want to see the whole community. And now it's more common a lot more common to see the extraction of either all of the DNA or all of the RNA component to do either metagenomics or metatranscriptomics. That's all due to the advent of sequencing and it being significantly cheaper and forever diminishing. And so really now the bottleneck is the analysis, in my opinion, and it's probably the most costly part of the whole process, barring some of the complex sample collection methods. So there's lots and lots of research going into metagenomics. So that's easy. So we just look at all of the different papers and we can compare them, right? Yeah, that's easy. Yeah, I can hear the sniggers in the audience, and that's because really there are so many different questions that people are asking. So if we just take the human gut microbiome, you know, if we this is just a random selection of articles I pulled out a year or two ago, and it varies from sort of looking at just who's there in a healthy state in terms of which organisms are present. Then there's question of can we return it to that healthy state when we've got C. difficile? What's the interaction between the microorganisms and the host? What happens when you're giving drugs? What happens over time course? How does it change? And the problem is that everyone uses a different approach, everyone uses different methods of analysis, and everyone's asking slightly different questions. So actually the comparison is nigh on impossible. And even when we look within a single project, so this is just a slide taken from this paper describing the American Human Gut Microbiome Project. And if we focus on, the American Human Microbiome Project, when we just focus on the gut sample, one of the things is that they've taken huge numbers of samples for the gut, and if we just look at the variance of the Bacteroides, it's huge. Now the question is this is just within one study, and we know that within this one study there's some variance, but they tried to standardize a protocol and they tried to standardize analysis. So is this real, or is this coming from the experimental method? So in this case we think it's real, but when you're comparing all of these different papers, it's very, very hard. So the EBI Metagenomics platform was set up to try and deal with this key issue, with this huge amounts of sequence data being deposited and no annotation being supplied with it routinely. So what we do is we take both public and private data and analyze it in a standard way, and we will work on anything. So we'll do amplicon, metagenomic, metatranscriptomic, and we'll take assembled data. And we've got a web interface that allows you to sort of browse, and you can descend through to your favorite biome. You've got ways of doing more complicated searches where you can search on things like depth, biome, you can look for different organisms, you can then look at, when you get down into individual runs, you can look at the taxonomic distribution, you can see output from our QC, you can look at the functional profiles of those, and you can do comparisons within a project. And we have two sort of real mandates. The data archiving is really, really important in terms of actually everything we do is brokered through to the European Nucleotide Archive. So that data gets submitted. Other resources don't have that long-term brokering. And we want to have this consistent analysis. So where are we in terms of EBI metagenomics? Well, how much data do we have? So we have, these numbers were collected 2 weeks ago, and these will be out of date if you go on the website, but we have over 1,000 different projects. Within those projects we encapsulate over 61,000 different samples, and that is more than 85,000 sequencing runs on those samples. On the right hand side there's a breakdown of how those sequencing on those projects, well, the sequencing runs what they're doing. So the vast majority, so 77%, is amplicon. We have a tiny fraction of submitted assemblies. We have meta-barcoding are things other than the standard SSU, so where they're using, say, COX-1 or ITS-1 to try and do similar sort of barcoding as amplicon. It's a sort of subdivision that we just use as an internal differentiation. And then we've got about 2% on metatranscriptomics and 20% on metagenomics, but that 20% is by far the largest fraction in terms of amount of sequence. And from that sort of data as a whole, that's over 250 billion nucleotide reads we've analysed. But when we start looking in terms of numbers from this metagenomes, we've got 3.6 billion SSUs that we've identified, 150 billion CDSs, of which about a third we can provide some sort of annotation on with InterProScan, and I'll come on to what that means, but that's some sort of functional annotation. Here is just a very quick slide to give you a sort of quick feel for the amounts of data sets and flavors and the approximate sizes. So this is in 2015 in December. So each one of these circles represents one of the metagenome shotgun projects. The size of the circle is proportional to the amount of data that's in that. So this big one in the middle here is Tara Oceans. All the blues are sort of aquatic or ocean. The reds are human associated. The sort of orange colour are sort of animals, and you've got, you know, soil around the outside. This does not include any of the things like the amplicon datasets, because they would just be tiny single pixels and you wouldn't see it. But there's a huge range of different shotgun projects that we have in the resource. We also have seen a lot of growth in terms of the amount of data we've processed over the last. So I arrived and we were sort of chugging along doing sort of 4,000, well, no, a couple of thousand data sets. Then we went up to about 4,000 or 5,000 data sets. And last year we nearly hit 60,000 data sets. A lot of that is due to us optimizing our pipeline, and some of that's also due just to the volume that we're seeing. We have, as I say, private studies. So we'll keep this private, similar to how people submit data into ENA. Their data is stored privately, you'll get it accessioned, then it comes back to us, and we can allow it to stay private for the time up until publication, and then it's released. And so this is why this grows at a relatively linear rate, because of the fact that data sets that were in 2014 have now become public. So there's really two parts to the submission process for the EBI's Metagenomics platform. The first part is you come to our website and then you're immediately brokered through to the European Nucleotide Archive, and that's the green part here. So once you've got your account, so this is a web-in account, you log in, you upload your data and get submitted. So then that's in the ENA. As part of this login process, you're asked to give EBI and Metagenomics access to your private data if you're coming down the private route. And after, it's less than a week, but after about a week, that file will be indexed and be available for us to start analysing. Now, there's 2 to 3 weeks here in terms of the analysis side. That's actually, for most projects, that's 2 days. It depends on the size of the project and how busy we are. So this is a question of managing expectations, but it's usually far less than that. So then the analysis starts. Once we've had that tick that it's been archived in the ENA, we then pull the files down and process it. And one of the key things that we get out of this process of using ENA is we capture metadata. And I'm going to talk more about that in sort of part 2. I'm going to focus just on the analysis side. So then, depending on whether you come in with public data, it either goes immediately open, or your private data can stay held for as long as you want, usually about 2 years' time. So here's an overview of the pipeline. So you've probably heard, so Niels, did you go through MetaPipe yesterday? So you've probably seen something very, very similar to this. In fact, there's been a comparison by Espen on the pipelines. They're quite similar. So but our pipeline, we take anything goes into our pipeline at the moment. So we can have amplicon, metatranscriptome, metagenomes, or assembled data. We take that reads or contigs. We'll perform some sort of QC on those reads, and then we go through. So each one of these boxes represents the tool. If there's a light grey box, that represents the reference database that we use. So we perform, if it's, for example, a Illumina paired end, we start off doing some merging. We then do some basic QC, so removing sequences with lots of ends. We have a length threshold of we want sequences over 100 nucleotides, etc., etc. Then we start off with the rRNA masking, where we use rRNA selector. They have some bacterial SSU or 16S, 28S and 5S library to then pull out the SSU and LSU, and then the SSU 16S. So I'll use SSU and 16S interchangeably. SSU is the correct term for it. So with our current pipeline that's publicly available at the moment, classification undergoes, the SSU undergoes classification using CHIME and green genes, and then we visualize with Corona. Although this shows a forking, this is really a linear process, because what actually happens is the rRNA goes through that classification, and then all of those regions then get masked. There's then identification of tRNAs using NHAMmer and just the model from RFAM as a prelude to expanding the non-coding RNA analysis. And all what comes out at the end are basically all of the reads with all of the non-coding RNAs taken out and masked out. So then those reads then go through open reading frame detection. We use FragGene scan there. Then we get past all of the predicted protein regions through interpro scan, and then you get your analyzed pot of sequences. We produce something called GoSlim, so gene ontology. I'll come on to what that is in a minute, but we have a slim version, and then we have the usual unannotated pot of sequences where we can't provide any information on what they are. And that's the current process. Simple. So what is Interpro? How do we do the functional analysis? So I think you've heard quite a lot over the last few days about CHIME and how that works. So I thought I'd focus on Interpro. So that's a resource that I look after in my team. So Interpro is a collection of 15 different member databases, and they all have a different view of what a protein family is, and they all use slightly different methods. And so here's a sort of classification, the ontology of protein families. So if we look on the far left-hand side, you've got the method. Then in the middle here, you've got what those databases are trying to represent, and then the individual databases. So as you'll probably see, the vast majority are profile HMM based, and at the top we've got the most diverse or broadest families, which are just trying to model sort of structural domains or almost folds. You've got Cath, Gene 3D, and Superfamily. We have then databases that try and model family protein domains, PFAM and SMART, and then you've got databases that try and model full-length families. So you've got TigerFam, PIR, SF, SFLD, and Panther. Similarly, you've got HAMAP and ProCyte profiles that try and do full-length families. Then there's CDD that tries to do domains and families, and ProDom. CDD and ProDom use RPS BLAST, whereas HAMAP and ProCyte profiles use PF tools, which which is out of SIB. Then there's prints, and then prosite patterns, and then we also have intrinsic disorder that's predicted by MobiDB. Collectively, what we do is that the sum of all these databases is far greater than their individual parts, because depending on your question, you can descend to different levels. You can try and pull out active sites, you can say what domains are found on a family, and there's a lot of overlap, so we condense equivalent families into single entries. Now, when the pipeline was first developed, what they did is they analyzed the differences of basically looking at the big hitters in terms of coverage. So to make the analysis go faster for scaling for metagenomics, what they've done is the team trimmed down the databases. So you've gone from this 15 to just these 5 with only minimal loss of annotation. So I should point out that each one of these grey dots also represents a separate post-processing step, if anyone's wondering what those are. Just another thing we have to sort of take care of. So for EBI metagenomics, we use this lighter version at the moment just for speed. And so why are so many of these databases using profile HMMs? Now, I am slightly biased, so I've worked on PFAM for 20 years or so. But it really encapsulates why it's important, and why so many protein family databases have used profile HMMs. Now, the way you build a family is very, very simple. So you start off with a set of sequences that are related. You then build a profile HMM, which just converts that alignment into a set of probabilities. It's not rocket science, so if you've got an invariant column, then the probability of seeing that amino acid is high, and the rest is seeing the rest of the amino acids is low. The key thing that it models as well are inserts and deletes, so that if you've got a gappy alignment like here, that gets modeled in this profile HMM. We search that against a sequence database, and we iterate until we can't find any new members. We have a carefully curated threshold in what we call a desk file or a description file, and some annotation. But the key thing of these profile HMMs is once you've seen a good fraction of sequences, they're very good at seeing things they haven't seen before. So what do I mean by that? So in this little anecdotal example, I'm going to show you an example of a profile HMM versus the nearest thing, which is Cyblast. And here what we're doing is we're going to look at the globin family. It's globin superfamily. It's well known, it's well characterized. So this is the structure of a myoglobin. Here's how the globins look. So if we focus on the top portion, we're going to start off with the alpha hemoglobins, beta hemoglobins, and myoglobin. And we're going to have 3 vertebrate hemoglobins and 1 myoglobin. We're going to make a little profile HMM out of those 4 sequences aligned, and we're going to use that same alignment to start off a Cyblast or a hammer search. So if you use Cyblast, what Cyblast pulls out are exactly what you've put in, so alpha hemoglobins, beta hemoglobins, and myoglobins. However, just an insignificant e-value, you can see some distant relationships with Cyblast. But using a profile HMM, you see not just what you put in, so you're not seeing just back that 550 million years of evolution, you're seeing back all the way, so you're seeing all of the neuroglobins, you're seeing plant leg hemoglobins, and you can see a very ancestral bacterial homologue, an e-value which I would say is like just about significant, and that's 2,500 million years of evolution. If you plot e-values, if BLAST and Hammer was the tool we use for searching profile HMMs, if they operating at the same significance, you'd be on this red line. What you see is Hammer sees things with more significance and picks up a whole bunch of globins that BLAST just misses. Hammer is also faster than BLAST. So why am I telling you all of this? Well, if you think about metagenomics, where we're going out into into an environment where a lot of the organisms we've never ever seen before, it's really important to have the sensitivity of the tool to pull out and see the distant relationships. So here we start coming onto the joys of running pipelines and some of the challenges and pitfalls. So hammer is now faster than blast. Great. It can also be very, very slow. So I'm not going to go into too much of the details, but hammer suddenly became much faster in 2010, and that was due to the introduction of a bunch of heuristic filters around the algorithm. The first filter of this is what we refer to as the MSV filter, and it's like a bit like an ungapped word filter. And so you're just looking to see if there's any chance that this sequence is going to match my query. Then the next one, it uses a profile HMM in a basic mode, and then using it in a super sensitive mode. When you're doing the analysis, 70% of the time of the algorithm actually is spent in this first heuristic filter. So when we were starting to analyze Tara Oceans, we released a new version to try and deal with. We upgraded the version of Hammer and libraries, etc. And we released it in March, and then we started maxing out. And it was maxing out about 2 million reads per month. And I was like, this should be much faster. That's currently going to take us a year to process. Back of the envelope calculation says this is completely wrong. And the reason is that essentially you can get very quickly bogged down in this. If you don't get the balance of the searching right, you get bogged down in I/O. So we put in a fix here, and then we shot up to 12 billion reads per month and managed to do Tara Oceans in 3 months of processing. Now, it's still I/O bound. We know how to make it go faster, but these are all the sorts of challenges that my team sort of deals with in terms of they're very good at finding these problems and making and coming up with solutions. Profile HMMs also give us some other benefits. So if you take a sort of typical profile HMM database of about 10,000 HMMs, and you compare that to what a lot of people do with, say, a BLAST database or a sequence database, and you want to compare that and say we've got 10 million sequences in that. If you look at the size of the data sets, so for a 1 million sequence input data set, that's 10 to the 10 comparisons for an HMM search, and 10 to the 13 for a sequence database search. It takes us to get to an input size of 1 billion until we're doing the same numbers of comparisons for a profile HMM. So if the cost of doing one of those comparisons is about the same, you can do significantly larger data sets in the same amount of time. And consequently, that's caused people to use inferior search methods like BLAT to try and increase speed, but they lose some of the sensitivity. So once our sequences have been annotated with Interpro, we also provide Go terms for all of those. So Go terms, or gene ontology terms, are a structured way of providing information that can be read by a machine about the function of the biology. And we don't maintain the Go terms, but there is a hierarchy associated with all of those Go terms. Now, there are so many different Go terms out there that there's thousands of Go terms. And they range from very sort of broad topics like signaling all the way down to more specific terms, and this is all of the relationships. But what we do is we have a Go slim, and what does that mean? So here's an example of a molecular function all the way down to its most fine-grained point. So you have iron binding, calcium binding, lead binding at the leaf nodes. And if we follow that up, we see metal binding, etc. So when we slim, what we do is we take all of these metal iron bindings and just roll that up to a slimmed iron binding term. So last year, the pipeline underwent an upgrade of the Go Slim. So the initial Go Slim was somewhat out of date. We could look through all of the annotations Interpro was providing and make sure the Slim was covering. It wasn't. Some 13% weren't annotated with a Go Slim, so we had to revise the Go Slim. So here you're just seeing the top sets of the Slim, and then there's others, and then there's a lot that are known. Here's the same data set on the old Go terms, and then the new data set where we capture now 97% of all of the function in this Slim, which is actually covered by only about 200 or so terms, making it much easier to digest the functional potential of the samples. We do, sorry, this has just slipped on the export, but so we analyze raw reads. How does that compare if you do assembly? So there's a Cow Rumen that's been assembled, yet we had the raw reads available to us. So we looked at the difference between assembled and raw reads. So HES is the paper where they've done assembly. Purple is EMG, and we're just looking at the carbohydrate metabolism enzymes from PFAM. And here, hopefully you can see there's pretty much perfect agreement, barring a couple of anomalies, and that's actually just down to post-processing differences that PFAM needs to have. So PFAM has this concept of clans, but essentially the counts that you're seeing are very, very similar whether you analyze the raw reads or whether you do assemblies. So there's lots of data. I'm not going to go through all of the different use cases for the website. Here's just some anecdotal examples of how people can use the site. So Yong Sound is a ocean sampling day data set. It's just off Greenland. So they took 2 samples, one surface water and one below the ice cap. And if we look at just a taxonomic profile, the one on the surface has lots of cyanobacteria, yet the one under the ice cap, which has minimal light penetration, has virtually no cyanobacteria. So we're seeing that from the taxonomy. Now this is a shotgun metagenome, so we can look at the functional side, and this is just an output from our comparison tool. The surface water is green, so the one at 0 meters. The one at 2 meters is blue, and if you look at the photosynthesis here, this just drops off the photosynthesis. We're seeing it from both the taxonomy and from the functional profile that photosynthesis is not present at the deeper sample. Other people download our data sets, and because we focus a lot on capturing metadata, they're now able to start working out, the relationships between our functional annotations and metadata such as pH. You can see certain families, salinity, temperature. Certain families are upregulated, others are downregulated. So one of the joys of running a very generic pipeline is, why aren't you using this tool? And here's just an example of one of the things that always annoys me. So an example that people frequently ask me is, well, why aren't you using Metaflan? Metaflan's great. So on the left-hand side, we've got an example of just using Metaflan in default way on a lean human gut sample. You get a really nice taxonomic tree coming out, covering virtually all of your data. If you do the taxonomic SSU profiling or anything, they all give virtually the same answer. However, taking an OSD sample, you see something that's pretty dire. And the reason is that Metaflan, it has been developed, it's got a reference set of single copy genes, and it's been developed for analyzing human microbiome data. And so it's very, very good, and those genes are well represented. When you come to data sets where they're more diverse and we haven't got such good reference databases, and I think you'll hear a bit more about some of these reference databases and how we're trying to address it from Niels. But the Metaflan, so we don't use Metaflan, it's just that it doesn't work across all biomes, and so we're constantly having to evaluate different tools and approaches. Here's another thing that we sort of see. So another marine example. These aren't just marine just because it's the only thing, but these are really nice examples. So here's an example where the submitted assembly to us, so you can discount analyzing raw reads versus assembly. This assembly is pretty good. So if you look at our analysis and you see this big chunk of missing data where we can't make any form of taxonomic assignment, what's going on? So this is from the Gulf of what's top of the Red Sea, this Gulf of Aqaba. Now the Red Sea's got some quite unique biology going on about it, because it's a sea that's actually growing. It grows about 5 to 10 millimeters a year in width, and it's got a lot of volcanic and seismic activity underneath, and so you get some quite weird and wonderful things happening. So we've only assigning 28% and so the of sequences to some form of classification. So what's going on? So we've tried to tear our pipeline to pieces to work out what the heck's going on here. So we've gone and got rid of the rRNA selector because it's not using the best tools. We've first of all tried running RFAM SSU models, so this particular RF11177 with NHammer to pull out the SSUs, and then through other work I haven't got time to describe is using using Silver as a reference database, because we believe that's the best, and a new tool that's not even published yet but is doing very well called MapSeq. Now NHammer is very, very sensitive at pulling out the nucleotide sequences, so we wanted to make sure, and this MapSeq gives you a confidence over your taxonomic assignment. So what we can do is, we first of all tried this on a human sample, and what you see here, so we've got the classification. Each box represents the most fine-grained classification we can get for a sequence. Along the x-axis is the negative log E value of the NHammer hit, so the further you are to the right-hand side, the more significant it is. Along the y-axis is the confidence supplied by MapSeq. Now for a human gut microbiome, you should see that there's very few points around here in these unclassified kingdom, phylum and class levels. Majority, you've got very high confidence of the order, family and genus, and even down to the species level. However, for that same Red Sea sample, we see this. And what you'll see, if you can actually notice, the vast majority of the points are in this kingdom level or unclassified or phylum. So we've actually pulled out more sequences. We pull out about 8,500 SSUs with NHammer in that model. But most of those are ambiguous, even using what we believe are the best tools out there. So how do we make sure that these are real? So we can use the secondary structure of the SSU, so we can actually go to the full covariance model. So this means that you actually look at the secondary structure. So what do I mean by that? So the SSU has defined secondary structure where there's base pairing between the sequence. That base pairing, for example, happens where these two red boxes are. That's the equivalent region. And that actually has no sequence conservation, but because the base pairing is kept, the Watson-Crick base pairing, you get a signal from that, and you can actually detect that, and there's tools for doing that. So you get about 17 bits from the sequence and structure compared with only 14 bits from the profile, so 3 bits extra from the structure. So you can actually use that to show that you're actually pulling out true SSUs. And so what we can do is we can take this information. So in this plot, what we've got are the bit score from NHammer along the x-axis, and then the amount of information coming from the secondary structure on the y-axis. And what I've plotted with this dashed line is an e-value equivalent of 10 to the minus 5. Anything up to the right-hand side of that line are definitely SSUs. And so really the take-home message is there's huge numbers of sequences and organisms we have no idea about what they are, what they're doing, because they just aren't present in the reference databases. And really that's strong motivation for going forward. We've got lots to learn. Human microbiome, that's easy. More diverse samples like soil or water are much harder. So all of this has stimulated us to start thinking about, well, start preparing our version 4 of our pipeline. So it comes to a point of playing spot the difference a bit, but the main thing is changing the whole classification here, and using MapSeq and Silver, and using more of Infernal and RFAM to pulling out more non-coding RNAs. So the other joys that we have to sort of cater for when doing metagenomics and maintaining pipelines is this constant surveillance of emerging technologies. So this is a slide I showed earlier. Actually, get rid of the size fractionation. But what's really changed here is we're beginning to see far more of different technologies. So here, we're beginning to see more samples coming through from Oxford Nanopore. And so we've looked at these, and it's interesting. So why is it so interesting? So this is a slide courtesy of Matt Clark, who works at the Earlham Institute in the UK. And basically, last year, Oxford Nanopore released a new chemistry. Matt's got it working pretty well, and he's sort of one of the sort of early adopters, so just take it with a slight pinch of salt. But you know, the R7.3, the error rate was about 85%. So this is for the template and the complement strand. If you do the 2D and merge that, you get about an 85% error rate on average. Obviously, there's smearing. But with the new chemistry, where they've really speeded up the analysis, or the flow through the flow cell, the template and complement, the template's much better, and the complement is somewhat worse. The reason for that is because of the speed of the DNA passing through the pore. But the 2D, where you do the merging with the template and complement, the error rate has gone to about 95%. Now, that's beginning to look interesting. However, things just fail miserably when you try and do anything at the level of proteins, but you can work on the DNA. So we're now having to, and this is very much drafted on my whiteboard in my office, and this is just a view of it. We now are thinking about how we're going to deal with the sort of functional annotation for Oxford Nanopore. We can use the first part of that pipeline, and that will work. So reading off the DNA, the profiles at HMMs are very fault tolerant, but we can't get good open reading frame predictions. So we're having to look at analyzing the DNA directly. There's also sort of changing sort of ways of working. So I said we worked always under raw reads, but now we're sort of moving over to the assembly and starting to think about that. Assembly tools have got a lot better. The depth of sequencing's got better. And so there's also lots of different technologies that are coming along to deal with longer reads, and we're thinking about how can we cater for all of these. So we've been profiling assembly. Basically, assembly's pretty easy with fairly sort of simple biomes. So human microbiomes associated, you can assemble that with very little, well, relatively little memory, about 50 gigs of memory, and depending on the input size, it just slightly changes, but the speed of processing happens within a day. With the other sort of aquatic and soil, I know there's not very many points on here. These are sort of very sort of preliminary graphs, but we've got more data points, but they still illustrate the same point, that they require a lot more memory. Soil especially requires a lot more memory to do full assembly on. Why would you want to do both? Well, typically with a soil sample, you'll only get about 1% of that data actually assembled out of it. But when you do get assembly, you get really good peptides out, full-length peptides. And this is one of the challenges in the fact that what people want, some people want to know just what's there in terms of that overview, but some people want to get access to the full-length peptides. And so we're starting to start building up peptide databases, and we hope by sort of September to start having some searchable peptide databases where you can actually look and see where certain sequences happen. Here's another version of the pipeline. So this is the assembly part. And I'm really building up to my next part, but we'll have a very brief interlude. So this is another version of the pipeline. We're constantly surveying and trying to have the best tools and the best techniques. There's still lots of unknowns out there. Now, when the GOS dataset was released many years ago, they thought there was huge numbers of novel protein families. There aren't huge numbers of novel protein families out there. There are some novel proteins. So they pushed a whole bunch of them through sort of structural genomics. So in the left-hand side, you've got monooxygenase. The blue is the template structure that is the closest relative in the PDB, and the grey is the structure from the metagenomic sequence. And I can see that relationship today. I could see it back then without any issue. It's just, if you're used to looking at sort of protein families, it just falls out. If you use profile HMMs, this relationship falls out. However, on this carbohydrate binding, the magenta is the best or the nearest relative in the PDB. The cyan is from metagenomics. I could never tell you from the sequence alone that that was a carbohydrate binding enzyme. There's just nothing there. It's just, it folds. So there is lots of interesting stuff out there. I think out of that dark matter, I think a third are just outliers of existing protein families. I think a third are probably just junk from the way that we analyze the data, and I think a third probably a novel, and that will obviously vary. That's a very general statement. Okay, and that really leads me to just thank all of these people who have been involved in some form or another. This is my team at the EBI. Not all of these people work on the EBI metagenomics, only 4 of those do. And there's the ENA team and a whole bunch of collaborators. And I'll take any questions at this point in time.